Mic check, mic check. And there we go. And then we're off to the races.
So like if you, alright, so if I see you on
Enemy soldier incoming. Prep time's over. Now you deploy to the war zone. Resurgence. Survive and your team can redeploy. Eliminate targets to bring back. Enemy dropping into the AR.
oh you beat body lit on that.
Back on station. Good work.
My kid. Yeah, I got one for it. How many you got?
Yo, my teammates, I need you guys to come with me. No, I got the key card. Come. We're going to fucking Stronghold key card specialist. Let's go. Guys, Stronghold specialist, fucking let's go.
Enemies are dropping into the area. Additional contracts online. Play. More risk, more reward. Target down. Marking the rest of them.
Where you at?
You've got a loadout drop inbound.
Fuck. always had to be a guy in the back of me. Fuck.
Get ready. You're redeploying. Mates are in the safe zone. You've got gas moving in. Enemy dropping into the AR. Contract failed. Remaining
train.
operators from that squad.
One more, one
Hunt
more.
them down.
Moving
We've
in.
got
Marking
4
enemy.
squads active in the field.
Marking enemy.
No, fuck off. Are you serious? He's telling me. Right here, one shot. Get out of here.
Advanced UAV. Loadout drop headed your way.
Mark it, I'm gonna land on.
You'll get another chance. Weapons free. Contract's worth more than usual, so get it done, soldier.
Enemy.
Fucking bitch ass. Yo, on me.
Enemy movement. Cancel that.
Go get your revenge. by for deployment to the war zone. Resurgence. Survive to redeploy fallen teammates. Kill to bring them back home. We've got 10 squads active in the field. Enemy squad incoming. Reinforcements inbound.
Yo, I got the card. Let's go to Stronghold.
Your squad mate is redeploying. Well done. Enemy UAV is on. Friendly loadout drops on the way. Additional target added.
Wow.
Gas is inbound. Enemies are dropping into the area. Watch the sky.
It's good to see shit.
Your squad mates back on station. Good work. Confirmed.
Right there.
We've located the rest of them. Squad mates MIA. We're reducing redeployment. Contract failed. Objective lost.
Bro, it's a kid downstairs.
Marking enemy.
Bro.
Taking a fire.
Enemy soldier incoming. Destroyed. Use your tac map to take down the rest of them. Get after it. You have reinforcements inbound. Target displayed. Bounty contract installed. Enemy dropping into the AO. 25 remain. Solid work so far.
Oh, I knew he was in the water somewhere. Fuck.
In the safe zone. Gas is closing in. We're returning you to the front. Earn this. The squad mate's back on station. Good work. Enemy deployed. Use your tac map to hunt down the rest of them. Enemy dropping into the AO. Enemy advanced UAV overhead. Fire sale is active. 5 station prices are recovered. Got an extra valuable opportunity here. Don't miss your shot.
I need medical.
Enemy UAV overhead. Get ready. Surgeon's window is about to close.
Fuck. This shit spot, man. I can't get it.
Gas is moving. Nice connection. You're redeploying. Watch the sky.
No matter where you go. No matter where you go.
Fire sale's over. Just in prices.
So,
We lost that one. We'll get a W.
it seems like it's running very, very slow. the fuck subscribe to me?
It's on their
Oh, it's
website.
on Twitch. I got you. Because I was gonna say
Yeah.
NASA live streams like every day.
Oh yeah, just supposedly from that streaming service. I don't know if they did like an interview. Let me see. Yeah, it says viewers will have the opportunity to hear from NASA astronauts live and ask questions about life in orbits.
Okay, can you see me and Dan? Hold on, let me skip this shit.
Sorry about the whole setup.
Yeah, my
process.
bad. We have a lot of had a lot of
Oh, you're
technical
fine.
issues. Are
You're
we good?
fine. Yeah,
yeah,
you
no
guys,
worries. Yeah,
you
just
guys aren't on cam though.
Oh, how do we turn?
I don't. Do we need you guys on cam?
I mean, they wanted to do it on cam though.
Yeah, we can do it on cam.
There you go.
Hello? And now do full screen.
There it is.
Yeah, there you guys are.
Good
deal.
Look at those two
Okay.
handsome devils.
Thank you.
Thank y'all.
I had to shower in
Wait,
like an hour.
Can you send Taylor a link to it? Like a link to the. Well,
What do you mean?
I should be able to do it. Like I don't know. Like texting it.
Honestly, whatever you guys have going on, it's good. Don't mess it up.
Okay.
Yeah, I'm not.
All right, are we good to go? Are you just trying to share with other people?
Mm-hmm.
Yeah, I think our family wanted to watch it too. I'll just text it to him really quick. Now that we got it figured out. Yeah, I'll share it with you.
Yeah, and we'll be sharing on the live stream as well.
Perfect. Oh, we should
yeah.
probably do the YouTube link. Yeah. Well, Dan, in the background,
Send it
I
to you
think
too.
just send
let him
Dan
get started.
a YouTube link.
You're
Okay,
good,
perfect.
you're good.
Here we go again. Okay. All right, I think we're good to go. Okay. So basically, I'm going to be presenting today on abiogenesis, and I study biology, and I've always been interested in this topic. When I was in 5th grade, I asked my 5th grade teacher, how did life begin? And she told me, oh, molecules wanted to create other molecules. So I was like, what? And then ever since then, I've been really interested in the topic. And yeah, I've been studying it for about 4 or 5 years now and compiling data. So does science suggest abiogenesis or intelligent design? All right, so biology is the study of life in living organisms. Cell biology is the study of cells in particular, which is what I study. And you can see right here, it's kind of at the fundamental center of all these other fields of biology. And cells are the basic and fundamental unit of life. Fun fact, the word cell means cellula, which is derived from a small room. So a cell is just a small room. Came from that. All right, so fundamentals of biology. I'm going to start by running you guys through a bunch of fundamental concepts, just so whenever I make a point on biology, you'll understand what it is. I'm going to give you some information on the cell and how it works and the central dogma, et cetera, et cetera. All right, so basically everything's made of atoms. They'll make up molecules, which make up these organelles inside the cell. Cells will make up tissue, organs, organisms, et cetera, all the way until you get to Earth. And characteristics of life are they are made of one or more cells, they have organization, they can grow and develop, they can reproduce, respond to stimuli, they need energy, and they can maintain homeostasis or equilibrium. All right, single-celled organisms are small yet very amazing. So over here is a dancing diatom called Bacillera paxillifera. And here's a picture of them that I took on the bottom. So this is just a little cool video I threw in here. And here are some diatoms that are arranged in a cool, unique fashion by Klaus Kemp. And yes, these are living organisms, and they look interesting because their cell wall is primarily made of cilia. And then over here is another little protist. So they're very complex and intelligent and amazing.
Didn't we find out like yours.
Yeah.
These little things are pumping back and forth, so they're like scooping up water. You know, like this is our digestive system in the middle, and they're just like going back and forth, just like pick up stuff to eat. I thought that was pretty cool. You know.
Really interesting. All right, single cells, small yet amazing. Over here on the right, here's a cool little video of neurons. This little guy at the top's like over here trying to make some connections. We have a human cell reproducing. In the middle is the chromatin, and they're being pulled by the microtubules, which we're going to look at a little bit later. And then we have a neutrophil, or a white blood cell, that's basically chasing down a bacteria. So yeah, here's some cool videos of like what we look at in biology, some microscope videos, etc. And then here's what I study, microtubules. They are the cytoskeleton of the cell. They provide shape, structure, movement, and sensory input to the cell. And a little fun fact about these guys is they are attributed to fiber optic cables in the neurons. So that's pretty interesting. And here's a video of it happening, like an animation, and then a video that I recorded of the microtubules. And basically I was able to track these because these microtubules have a fluorescent protein at the end of them, so I can track them growing. So yep. All right, and then now we're going to move to intercellular components, organs. So they have a lot of them. They have the cytoskeleton, which is what we just looked at, different types of them, the microtubules, etc. They have the cytoplasm, which kind of fills up the inner cell. Let's see, chromatin in the nucleus. It holds like the DNA. And then you have the endoplasmic reticulum, rough ER. This will make proteins, and then smooth ER makes lipids and stuff like that. You also have the mitochondria right here, which you can see on this electron micrograph. And then also we have the Golgi apparatus, which you can see. So I know a lot of people look at this diagram and they're like, that's really interesting. Like they're a little skeptical sometimes of things in the cell. But these are some pictures of them that have been taken. And yet they're very complex and amazing. It's kind of the point I'm trying to hit home here. And this is a fun fact about this picture. It was made by Dr. McGill, and he's the director of molecular visualization at Harvard Medical School. And he said, although people are struck by the complexity and density of this environment, what we depict is actually very dilute in its concentration relative to a real cell. So this is a dilute form of a real cell. So
you can see the density in those electron micrographs.
Yeah. Well, there's a lot going on in there, and it's all needed for life. All right, now cellular composition. The cells are made up of 4 different types of macromolecules. You have the proteins, which are made of amino acids, the lipids, which are made of fatty acids, carbohydrates, those are just sugars, and then you have the DNA and RNA, which are made of nucleotides or nucleobases. And all types of macromolecules are chiral. So like the proteins, for example, they're left-handed, the lipids are mostly right-handed, carbohydrates are mostly right-handed, and DNA and RNA are right-handed. So yeah, all of the macromolecules are chiral, whether they are left or right-handed. Yep, still chiral. And so this basically means chirality means that they can't be superimposed on one another. So like if you take our hands, for example, you cannot superimpose them on one another. They're fundamentally different. So you can see the little depiction right there. And the purpose for homochirality, it helps the molecule basically have structural integrity, and it will preserve its function. So chirality is very important and necessary for these macromolecules. All right, then the molecular composition of cells. This is just to give you an idea of how big they are and how diverse they are. So they are made of 100 billion atoms. The smallest cell, mycoplasm, that's a bacteria. It's made of 100 billion atoms. One yeast cell has 42 million proteins. The shortest DNA has 580,000 base pairs and 507 genes. And this DNA would be 34 million atoms, just to give you an idea of scale.
That's to make the point that it's not like just by on accident that all these genes and the DNA is necessary. Like these things are necessary for life. Like it's not going to be like replicating and choosing to replicate big long strands of DNA and generating proteins if it wasn't required.
Yeah, it is basically necessary. And then here's some molecules down here that are required for all life, some different elements, oxygen, carbon, hydrogen, etc. All right, so now we're going to get into a little bit of functionality before we go into abiogenesis. So how does it work together? How does it store information? How does it use it? So cellular information system. So this is the central dogma of biology. Basically, information goes from DNA to RNA to proteins. And this is essentially the central dogma. So the DNA will be transcribed into mRNA that will then give the information to be generated into a protein. So then, yep, you basically have that transfer of information going on here that's required. and then the proteins actually make the DNA. So, yeah. All right, so Francis Crick was the one that termed central dogma, and the term dogma was actually a misnomer, as it implies an unchangeable belief which contradicts the scientific method. And Crick himself later acknowledged this improper use of the word. So it's not a dogma. Everything is up for testing in science, and we should be able to test and to validate things, and not just say everything is concrete. You should also be using the scientific method. All right, and down here is a picture of E. coli's DNA, and you can kind of see that double helix going on there. All right, and then here's a short video just to show you like the cellular machinery required to reproduce these molecules and help you understand the central dogma. So it's like less than 2 minutes. Let's see if I can speed it up.
The DNA double helix contains 2 linear sequences of letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain sneaking out the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Cool.
The grid you can see in this microarray.
All right, so you can kind of get an idea of the machinery that's required for cell replication and protein synthesis.
Yeah, that was awesome.
The grid you can see.
Okay. All right, so now we're going to get into origin of life. Dun, dun, dun. Just kidding. All right, so was it a random cosmic coincidence? The scientific community hypothesized that life originated through a random, undirected process known as abiogenesis. And abiogenesis is the idea that life arose from non-life. And here's a fun fact. Panspermia is the concept that life may have originated elsewhere in the universe and reached Earth through space travel. Watson and Crick proposed after discovering the shape of DNA. This begs the question of how it began. So basically, they saw the shape of DNA as a double helix, and they said, wow, this is amazing. And then they said, oh, well, they thought it was designed, essentially. But they couldn't say it was God, so they said it was some extraterrestrials, possibly, that brought this DNA over. So just a fun fact. But if anybody does bring up panspermia, just ask them, where did life come from there? Wherever it came from in the universe, how did it originate there? You're just pushing back the problem. Not to mention in the vacuum of space. Right. All right. So I'm going to go over the history of abiogenesis. So in 1859, Darwin proposed the theory of evolution in his book On the Origin of Species. The term abiogenesis was coined in 1870 by Thomas Huxley to describe the process by which life arose from non-living matter. And then Darwin in 1871 described the warm little pond that life could emerge from. And then we got the Operon and Halide hypothesis in 1929 that said life could have arose from a primordial soup, a mixture of organic compounds in early oceans energized by lightning or UV light. And so right here is just kind of a depiction of some of the things they think could have happened. So you have this abiotic synthesis, synthetic reactions on the Earth, right? Like making these organic compounds, and they have these organic compounds coming from outer space into a prebiotic soup to make these polymers, and then these polymers somehow just made life. And then we have the RNA world, which we're going to get into later, and protein DNA. These are some different. Some people think protein and DNA created life. Some people think RNA created life. Some people think amyloids or proteins themselves alone created life. Other people have metabolism first hypothesis. So there's a bunch of different hypotheses, but
Either way, if in any of those other hypotheses, life has to completely change in order to start working in a different way, like other than what we see today.
exactly, 100%. Yeah. So we're going to go over some of their theories a little bit later. All right, so I just wanted to give you guys an idea of how much they knew about the cell at this time. So at the time of the proposal of abiogenesis. So in 1868, this is the Fortnightly Review. This was a popular magazine in 19th century England. Okay, and this piece was titled Mr. Darwin's Hypothesis. Okay, so it's basically talking about the cell and the idea of the cell at the time. So I just highlighted this part. It says a cell is a nucleus with surrounding protoplasm. So that's what they said the definition of a cell was. And before this time, they did not have micrographs like at this time. So they were not able to see all the intricacies in the cell. And they just thought basically the cell was just some ball of jelly and a nucleus. So, yeah. All right. And then abiogenesis continued. It was 70 years after abiogenesis was proposed that we can actually see the intricacy inside the cell. So here's some pictures of inside the cell through an electron micrograph. You have the mitochondria over here. You have the cilia, and etc. All right, so in 1940, Manfred von Ardeen constructed the electron microscope, which is the reason why we're able to see these things.
Click on
All right.
shoot.
All right, so I was looking into the current state of abiogenesis because there's so many conflicting hypotheses that I was trying to see, okay, well, if I go get a biology textbook, I wonder what they're going to say about it, right? So basically the only thing they mentioned was the Miller-Urey experiment, which we're going to get into next. All right, and so this is basically one of the first and most prominent experiments for abiogenesis. It is the most cited evidence for abiogenesis, so we're gonna go into it. All right, so you have Miller and Urey over here on the right, and they attempted to simulate early Earth's conditions, and basically they were trying to see if they can make organic compounds, right? So they mixed the gases methane, ammonia, and hydrogen in a sealed chamber, and then they added water and heat to create vapor, simulating the oceans. Then they applied electric sparks representing lightning, and then they cooled and condensed the vapor representing rain or condensation, and then they collected and analyzed the tar that was produced. And over here on the right is what looks like the tar that was produced. So it just kind of looks like a dirty bong, but this is what they analyzed. All right. Okay. So here's the Miller-Urey results, right? This is a paper chromatography, and it's showing these 3 amino acids that they found. So they found, let's see, it's hard to read, aspartic acid, glycine, alpha-alanine, and beta-alanine, but I don't think aspartic acids. So just these 3 right here. All right, and so basically these amino acids, these few amino acids, composed only about 2% of the solid residue that was formed from the experiment. And then the amino acids that were formed were a mix of the left and right-handed forms of those amino acids. So remember earlier how I mentioned chirality and how they all have to be homochiral, facing the same direction? Well, in experiments like this, they get heterochiral amino acids that would not be able to form a protein anyway. Oh, yeah. All right, so this is just the reaction that occurred. It's not that impressive to me. They basically just used methane, ammonia, and hydrogen. They shocked it, and then they made glycine, beta-alanine, and alanine. And these compounds are only like 10 atoms. So we got glycine, 10 atoms, beta-alanine, 13 atoms. But don't forget, the smallest cell is made of 100 billion atoms, just as a reference and comparison. All right, so now here's some criticisms of Miller-Urey. So this one is by Dr. Scott M. Hughes, and he basically said that the concentrations of the components used were basically carefully selected, which would not be like that in prebiotic Earth conditions. It would just be haphazard. molecules. And he said there's no evidence to indicate the Earth's early atmosphere was reducing. And a methane ammonia reducing atmosphere would be fatal. Lightning would have destroyed the organics. He basically said the molecules produced from the experiment would have been detrimental to life forms, that tar that we looked at. And then he said they separated the amino acids from the mix, and then the chirality issue with the amino acids, and then formaldehyde and cyanide are destructive to life, and these are two of the things that were produced.
Exactly. So I just want to mention that this reducing atmosphere and the formaldehyde and cyanide are two things that you have to do, and if you want to make these nucleotides in the lab, like in a test tube like this. However, you see how two different examples of where they would be either, fatal or destructive to life.
Right. To make these amino acids, absolutely. All right. So some scientists speculate that they did not imitate earlier conditions properly. So here's one quote. It is possible that the primitive atmosphere was not as reducing as Operin and Urey and Miller had believed. Another one, the accepted picture of Earth's early atmosphere has changed. It was probably O2 rich with some nitrogen and a less reactive mixture than Miller's, or it might have been composed largely of carbon dioxide, which could greatly deter the development of organic compounds. And then some models suggest that there were significantly lower amounts of NH3 in the oceans at the time. So and also at the time when he was doing this experiment, there were other experiments being conducted that were similar to his, except for they were in an oxidizing environment and they basically couldn't get anything. So if it was oxidizing or even neutral, then you wouldn't have got what he had gotten, which is just a few amino acids.
And after reading some research articles about what early Earth conditions were most like, it would have found that it would have been highly oxidizing. Because if we believe that there was a lot of carbon dioxide in the environment, that eventually makes hydrogen ions, protons, and bicarb, and so it makes the environment much more oxidizing, not favoring the creation of these
organic molecules. Yeah, 100%. All right, so in their book, in Urey's book, The Planets, Their Origin and Development, it stated the primitive oceans would have been approximately 10% solution of organic compounds. This would provide a very favorable situation for the origin of life. So just to give you a reference, right, the organic matter in our current ocean at the sediment is approximately 1 to 3% organic matter. And they're saying it would have been 10% organic matter on early Earth. So kind of a stretch. All right. Here's a quote from Dr. Albert Bransom. He says, we claim that living systems evidence a preoccupation with maintaining chemical specificity, not just having the right molecules to hand, and especially not having all the wrong ones. Yet in any solution chemistry or any chemistry not managed by a macromolecular machine acting specifically to affect the particular transformation needed, the latter wrong ones invariably overwhelm the former right ones. So remember earlier how I said they only got 2% amino acids out of all that gunk and tar that was produced? So that's basically what he's addressing here. All right, and then now we're going to get into a couple problems, right? The tar problem. So the building blocks assumption is simply dead. This is actually by an original life researcher, John Sutherland, and he's made a lot of prominent papers on original life. So he believes in abiogenesis, but he is conceding this point. The building blocks assumption is simply dead wrong. As a simple thought experiment should suggest if you make up any mix of building blocks products, if you make up any mix of building blocks products you like, however rich, energize it in any way you like, wait as long as you like, nothing we assert having any claim of any sort to being lifelike will ever arise in your flask, not ever. Instead, the chemistry therein will more or less slowly progress towards a Benner's tar. Right? So whenever you have these compounds and you can shock it, you can do whatever you want to it, energize it however you'd like. All we keep getting is this tar, and that's definitely not conducive to life. That's the point. All right, so here's my search for a concise abiogenesis model, which was a lot harder than I originally thought. So I just pulled this from a paper that was published a few months ago. And basically, they were just using a computer code to infer the order of the addition of amino acids. So I was looking at it to see basically where we're at in the original life research. So this was published only 3 months ago. Here's some highlights from it. That the modern genetic code was assembled in stages, hypothesized to begin with early amino acids present on the Earth before the emergence of life, possibly derived by extraterrestrial sources such as asteroids or comets, and ending with a late amino acid. So they are definitely invoking these extraterrestrial sources and comets and meteorites, etc. It's so funny. And then they talk about the hydrothermal vents. They talk about the cyanosulfidic environments that they're having to invoke. They said amino acid abundances obtained from asteroid samples will also soon contribute. So they're waiting on these asteroid samples to see if they find more organics in it. That's so funny. And then they also reference one of Saturn's moons here. And I can't say this word. Can you, darling?
Enclidius.
Enclidius's moon. Yeah. So anyway, and then the hydrothermal vents and then tRNA synthetase, they're kind of wondering how it translated everything into the genetic code as it is. So we'll get into that later. All right, so here's a little summary I made after looking at dozens of papers on abiogenesis on where we're at, okay? So here's the storyline, get it. Between the edges of a hydrothermal vent, ocean water, and a volcano rock pores form nature's chemical laboratories. Each pore offered a unique microenvironment, some stockpiling amino acids, others stockpiling nucleotides, while a few fostered the assembly of RNA strands. Lipid components delivered by meteorites crashing into the Earth provided raw materials for primitive membranes. Lightning strikes while UV light radiated shallow pools, driving critical reactions. Meteorites crashed near these pools, delivering essential elements and providing energy for certain chemical reactions. Clay surfaces acted as a molecular assembly line, aligning and concentrating molecules in a poured rock near a hydrothermal vent, while magnetic minerals aligned them into just the right configurations. Furthermore, relentless wet and dry cycles in these pools alternately dissolved and concentrated molecules, forcing them to link into more complex forms. Eventually, RNA migrated from the shallow pools to the hydrothermal vents. Tiny lipid bubbles encapsulated the RNA while being able to hold the contents and transfer materials inside and outside of them without breaking down, and began migrating between these environments, scavenging compounds and energy wherever it could. Through this interplay of hydrothermal vents, meteorites, lightning strikes, UV light, and drying pools, the first cell was born. Voila.
Wow, what a story.
Man, crazy. All right, moving on. So basically, I made this chart to try to tell you guys just a few of the conditions and environments that would have needed to be present for the first early organic compounds to arise. Okay, so we got this matramorolite clay that is needed for the formation of RNA. And the environment would be the tidal pools or volcanic ash areas. And then we have the pyrite, which would be needed for the pyrophosphate synthesis. And this would have happened near hydrothermal vents. And that's different than the tidal pools, by the way. And then you have you have this iron sulfur minerals needed for amino acids and redox reactions. This would have had to happen near hydrothermal vents. And then the borate minerals would have formed the sugars, like formose, ribose stabilization, and this would have happened in a evaporative environment. I'm assuming in a pond or whatnot. So anyway, you're just having to see like how they are invoking all these different things for all these different cellular components, but it doesn't really work well together. That's kind of something that we're going to see soon. So here's some of the conditions that I saw when reading these papers, and I just basically did bullet points of them. Okay, so we got these wet, dry cycles. We have the lightning, the UV light, the clay, the comets, the meteorites, the hydrothermal vents, the shallow pools, the porous rock with subsections of environments, by the way, and crystals. So, yeah. All right, so here, this little graph is also showing incompatibilities between the different molecules that would have needed to be formed. So you have the DNA synthesis at a nice 55 to 85 degrees Celsius. And then the protein synthesis would have needed to be greater than 100 degrees, right? So DNA and RNA synthesis would have also needed UV light, but protein synthesis was basically hypothesized to be formed in a hydrothermal vent, not using UV light, just using heat. All right, and as you see, they need much hotter environments than DNA and RNA. And then, let's see. Oh, the proteins require low water activity. The RNA requires wet, dry cycles. And then you got the DNA that needs to be in an aqueous environment. But if the water is like contaminated with too many organics, it's prone to hydrolysis, or basically it'll just disintegrate and fall apart. And let's see if there's anything else. Oh yeah, so the shallow ponds for the DNA and the RNA, then you got the volcanoes for the protein synthesis. And here's some quotes from original life researchers actually contradicting each other. So it's saying a requirement for UV light to generate biological building blocks would rule out the deep sea environments or the hydrothermal vents, assuming that they're deep sea hydrothermal vents. But here's another one. To get one's electrons by UV photons at the cost of exposing a tender and unprotected neonatal metabolism to a notorious destructive radiation, UV, we would like to offer a much better deal, hydrothermal vents. So they're contradicting each other. One is saying we need UV light, one of them is saying we need hydrothermal vents. So
And there's a bunch of incompatibility, and we're gonna get to more of the paradoxes coming up.
Here's the incompatibility paradox. What do you know? All right, so the Maillard reaction. Amino acids can react with sugars like ribose, a component of RNA, in a Maillard type reaction, which could deplete the available ribose for RNA synthesis. So you can't have amino acids and these ribose sugars needed for RNA together, but somehow they would have needed to be together in order for the first life to form. So they must coexist, but they can't be around each other. So get that. It's science, right? All right, so here's a translation paradox in terms of magnesium. So the tRNA folding needs a 15 to 20 millimolar of magnesium, whereas nucleotide synthesis would need less than 2 millimolar. And then you got the RNA bonding that has 15 millimolar total concentration, and it can maintain stability at around 2 millimolar, right? So
I just want to mention how here you said like less than, but it says like great.
Oh,
If it's
greater than,
greater
thank
than
you.
2, it'll suppress its
Yeah.
incorporation.
So if you have greater than 2, right, millimolar concentration of magnesium,
it
it'll
does
suppress the nucleotide incorporation in DNA.
Mhm.
However, in these other cases, you need much more RNA. Much more magnesium, like 15 to 20 millimolar. So this is an inconsistency right here. But basically, the cell will do different things to separate these and incorporate form, and that's one of the amazing examples of the cell. All right, then you got the phosphate paradox. DNA and RNA synthesis. So phosphate has a positive role. It's needed for the activation of nucleotides during DNA and RNA polymerization. However, free phosphate groups, which would have needed to be there for the first cell, such as 3-phosphate on nucleotides, can block DNA synthesis and prevent polymerization activity. And then you have the amino acid and protein synthesis. They have a positive role. They're necessary for the activation of amino acids like aminoacyl tRNAs. However, they can interfere with the ribosome function, so excess free phosphate will disrupt ionic balances that are critical for ribosome assembly and function. So you need some free phosphate, but if you have it just open in the environment, it will mess up other things, right? So just some incompatibilities. And then here's another fun little phosphate problem. So bioavailable phosphate was required for energy production and as a component of the DNA backbone. However, phosphorus containing minerals on early Earth, such as apatite, calcium phosphate, were highly insoluble. So they're like, okay, well, we need this phosphate, but all the phosphate is bound up in these rocks, right? So that was kind of a huge problem. And then NASA came out and said, oh, wait, we don't need phosphate. We found this bacteria that can incorporate arsenic into its DNA backbone. And then they published that paper, and then basically a lot of scientists kind of squinted at it and was like, what? And so some people tried to reproduce it and ultimately found out that the paper was highly flawed. There was a lot of errors in the experiment, and it was just straight up fraudulent. So thanks, NASA. Yep. All right, moving on. So the water paradox. So water is commonly viewed as essential for life. So are biopolymers like RNA and DNA and proteins. However, these biopolymers are corroded by water. For example, the hydrolytic deamination of DNA and RNA nucleobases is rapid and irreversible, as is the base catalyzed cleavage of RNA in water. So this allows us to construct a paradox, right? RNA requires water to function, but RNA cannot emerge in water and does not persist in water without repair. So these are why our cell has these mechanisms in order to maintain homeostasis with everything. So it'll have what it needs, but then not be degraded by it as well. All right, here's an energy paradox. So energy is needed for life. However, raw energy without proper ways to process it will cause destruction. It's like trying to charge your phone using a steam engine or a charcoal grill. It's like you throw your phone in a grill, it's like, yeah, I'm charging my phone. Well, what's gonna happen? That cell phone is just gonna basically degrade. Or it's like trying to put a Tesla charger in a gasoline intake hole of your car. If you don't have the right energy and you have no way to process it, it can be harmful and destructive. And was there something you want to say about the energy? It's okay if not.
No, just that if anybody has any questions about why some of these reactions are impossible, we can talk about delta G for some of the reactions later.
Mm-hmm. Yep. All right, so here's a UV paradox. So UV light is crucial for the formation of key organic compounds necessary for early life forms. However, it is destructive, right? So excessive UV radiation can actually break down and destroy the very molecules it helps to create. How sad. So yeah, it'll cause what's called a pyrimidine dimer in DNA. So they're invoking it for the energy and the formation of it, but wait, if it's exposed to UV light too long, it totally degrades. So cool story, bro. And the hydrothermal vent paradox. So hydrothermal vents, as you guys saw earlier, are proposed for protein synthesis. However, a study published in Nature Geoscience found that hydrothermal vent conditions dissolved free organic compounds, carbons, into carbon dioxide. They wrote that even the most resilient organic molecules eventually broke down as they circulate through the vent systems. So, yeah. Hydrothermal vents, while they could be necessary, can be very detrimental in some cases. And that's why they have to have these, rocks with pores and wet dry cycles, and it's just a lot they have to invoke. All right, the heat paradox. So heat is a catalyst. It'll accelerate chemical reactions and provide energy for forming complex organic molecules. However, heat is also destructive and can break down delicate biomolecules such as RNA and proteins. So yeah, it can be helpful and harmful. But remember, in early life, you only had one condition it had to form in. They all had to come together and form somehow, right? So this poses a paradox. How does that work? All right, so now we're going to get into some of the cellular paradoxes, the DNA protein paradox. So modern cells require protein to replicate DNA and RNA, but proteins themselves are encoded by these genetic materials. This creates a circular dependency where the components necessary for life to rely on each other to exist, complicating the understanding of how life could begin from simple organic compounds, right? So basically the same thing that makes the DNA is the same thing that is encoded by, right? So DNA makes proteins, gives the instructions for proteins, and proteins actually build the DNA. So which one came first? And so to overcome this, they're trying to say that, oh, well, RNA came first. It's like the mediator, right? Well, if we look here, the RNA, the proteins are also used to produce RNA, right? So, and the RNA gets its instruction from the DNA. So it just kind of goes in a circle and we can never pinpoint. And even if you did have some self-replicating RNA with no machinery to do anything with it with, like it would just be some self-replicating RNA. It's not giving code or instruction for anything, so it wouldn't eventually move to life unless you had other systems already present to do something with it. You need to do something with that information. So, yeah. And that's assuming that all of the RNA molecules, all the nucleotides came together in the right fashion, all in the correct chirality and in the proper conditions. And you're having to invoke a lot to even give them that. All right, so here's Eigen's paradox. So we can't have long strands of DNA unless it can repair itself. And we can't repair itself without long strands of DNA. So this poses a paradox. So here, this was posted in Nature, actually, published in Nature. It says, he posed the paradox that without error correction enzymes, the length of a nucleic acid would be limited because in larger molecules, mutations would destroy the information content of subsequent generations. But this maximum size or error threshold was too small to encode an error correcting enzyme. Eigen's paradox still challenges theoretical biologists, LOL. And I love how they said theoretical biologists. Yeah. Basically pointing to the original life researchers. So anyway, that's funny.
Fairy tale science.
Oh my gosh. Okay, so over here is Eigen, Manfred Eigen, and he's giving a little thumbs up like, hey, try to figure that one out. So yeah. All right, then we have the membrane paradox. So the membrane formation paradox acknowledges that cell membranes can only be synthesized from pre-existing membranes. As an extension, the organelle formation paradox states that the membrane encapsulating organelles can only be produced by pre-existing organelles. And then the protein phospholipid paradox demonstrates that the protein composition in organelles is dictated by the organelle's phospholipid composition. But the phospholipid composition is dictated by the protein composition. So where do we start? Where's the starting point? How do we get past this?
You can't.
It's also interesting to have a membrane basically encapsulate this primordial cell, whatever that is. So the membrane would have to basically allow things to come inside and out of it. Because if you just had a little lipid membrane encapsulate it, basically nothing is moving in and out. You're not going to get any more nucleotides to make any more RNA. You're not going to get any more amino acids to make any more proteins. So that's why the cell has transport membrane proteins already in the cell membrane to move things in and out without having to degrade that cell. So, yep. All right, ribozyme paradox. In order for the ribozyme to be functional, it must be stably folded in a complex structure, much like a modern-day enzyme. However, in the absence of a set-aside genome, this same ribozyme would also need to serve as a template for its own replication. Templates cannot have complex stable folding if they are to be accessible to free nucleotides and RNA primers. Clearly, the same RNA cannot meet these two mutually exclusive requirements simultaneously. In the RNA world hypothesis, ribozymes necessarily must have these dual roles. They must have catalytic activity while simultaneously being able to act as a template for replication. Catalytic activity would require stable folding into complex structures, whereas its role as a template requires unstable, relaxed folding. So you can see it over here. The ribozymes, they must have catalytic activity, which requires stable folding. The ribozymes also must carry their own genome. To be replicated, they must have templating activity, and that requires unstable folding. So does it have stable or unstable folding? So the ribozymes, basically, with the RNA world hypothesis, they basically say that these RNA strands, right, double-stranded RNA strands can basically have these catalytic functions, but in order for it to replicate itself, it has to be in a folded manner, right? But in order to get its template, like its information to replicate itself, it has to unfold to give that give the nucleotides, make them accessible basically to come copy. So it needs to be folded to replicate itself, but unfolded to get the information from. So, how does it replicate itself? That's a big problem.
And if someone would say that this thing can fold and unfold like without a problem, they would not be understanding folding correctly. Because like when something folds properly, like it usually takes like other proteins called chaperone proteins to go and like pull on them and get them to like fit right. So folding itself is a big deal.
Right. And so there would have to be two simultaneous ribozymes be created, or one ribozyme and then one functional RNA strand that actually meant something. And then even if you had that, it'd just be continuously replicating this RNA strand, if giving them a lot, right? So they're just replicating this RNA strand. Then what? So how do we get to DNA? How do we get to a protein? Is this encapsulated by membrane? Does that membrane allow for things to transport in and out of it? So even with this is one of their newest findings, this ribozyme, they can self-replicate. That was a huge deal. So, but even with that, there are significant problems. And in my opinion, you're still not getting anywhere close to life. All right, circular synthesis paradox. So basically in the cell, in certain cases, you need something to make something, right? So you need 6 ATPs to make 1 ATP, to make any ATPs, right? You have to have them already there. So it takes NAD plus to make NAD plus. It takes tetrahydrofolate to make tetrahydrofolate. It takes coenzyme A to make coenzyme A. Same thing with FAD. You need it to make it. So here's the citric acid cycle over here. And yeah, for a lot of these compounds, like they already have to be present to be made more of. So poses a huge problem. All right, then the tRNA paradox. The translation system requires a highly evolved set of RNAs, proteins, and enzymes for high fidelity. However, these proteins could not have evolved without an accurate translation system in place, creating a chicken and the egg problem. So basically, let's see, tRNA is on here somewhere. So basically, tRNA is what is the translator for proteins and RNA. So basically, the tRNA will basically match up the nucleotide sequence on the RNA to the amino acid sequence needed for the protein. So it's kind of mediating between these two languages. So if you just think about it, if you had some amino acids in a protein and if you had some nucleotides in RNA, what's going to mediate in between them? Like where is the language going to come from? Like the translation going to come from? It's like sitting in a room staring at a computer code, right, on a piece of paper. Like, do you think that over time, eventually you'll understand what those ones and zeros are saying? No, you need a translator of some sort. So that's where the tRNA comes in. And that would also have to be there. And it's very difficult. You'll see the tRNA later, how complex it is. All right, so natural selection paradox with Charles Darwin. So abiogenesis will appeal to natural selection to get complex machinery. However, you cannot have natural selection until you have self-replication, which requires complex machinery. So where is this natural selection going to come in at? Chemicals can't naturally select. Chemicals have no goal or function. They're inanimate molecules. They have no end in mind, you know? So natural selection isn't going to help you there. And then if there is some sort of, you know, strand of DNA or whatever, in order to have natural selection, you need these this machinery to replicate it and to get feedback. But if that machinery is not there, you cannot get natural selection, right? You need this whole system to be in place to even be able to start choosing, you know, which nucleotides we want or which cells live and which ones don't, in order to continue on the ones that live, etc. So without this machinery there and without the system in place, you can't have natural selection. So that would come in later. Right now, we're just talking about chemical evolution, even though chemicals are totally inanimate and have no end or goal in mind. All right. And then lastly, the time paradox. So complex organic molecules require time to accumulate and become useful through random chemical processes. However, time is the very thing that will degrade organic molecules on an early Earth. So given enough time, anything can happen, just doesn't cut it. Because over time, these precious organic molecules that you just made will totally degrade when exposed to forces, the elements of, yeah.
And you hear this
nature.
argument so much from people.
Yeah,
I would just
with enough time,
billions and billions of years, you know. It's like
voila, it's there,
Yeah.
you know.
it's a fairy tale.
In many cases, it will just totally degrade these molecules they work so hard to produce. So moving on. All right, so this is a funny quote. RNA world hypothesis, the worst theory of the early evolution of life, except for all the others. So this is just funny. There's significant problems with every theory, and that's kind of what I'm trying to hit home here. They can't even come together and decide what came first. Some people say amyloid first. Some people say RNA first. Some people even say metabolism first. So they can't come together because there's problems in each. And so, yeah, it makes it hard to make one cohesive theory. All right, so British origin of life researcher John Sutherland concedes to the lack of progress in explaining a naturalistic origin of life. So it's funny, in this diagram he made down here, and if you could see the little purple in the bottom, it says the current state of the field. So we have this inherently favored prebiotic chemistry, then we have like energy dissipation, major system innovation, optimization of major systems, and then evolutionary innovation up here.
Like all that has to happen before it even gets
Yeah.
above the alive
Yeah,
line.
you see the alive line. All this has to happen before we even get to this fully alive line, right? And he says we are here, basically at the bottom. So he concedes it, and this is a prominent origin of life researcher. Please remember that. And I argue that we're actually back here, not even on the chart. Like we're below zero because actually the more we learn about the cell, the more we see how impossible it is. The more that we learn about all the machinery required for each of these processes and all the conditions each of them would have to be in order to form, the more we learn, the more we realize how could this have ever happened. So that's why I argue that we're way back here. Not to mention the characteristics of life, which I kind of ran through earlier, but living cells need to have a lot of things in order to function. And if you want to say, oh, well, the cell, you could just have like a very simple cell, you know, even a simple cell would still need replication. It would need some form of energy. It would need to translate to carry out protein synthesis. It would need to have a translator. It would need to have somewhere to store this code. It would need to be encapsulated by a membrane that could somehow move things in and out of the cell. And then eventually you got to start making your own nucleotides because, I mean, if we're encapsulated, how are we going to make that work?
They need
to get more. You need
membrane.
Constantly new materials coming in, and I'm not sure if their membrane would allow for that without degrading. So anyway, the more we're learning about the cell is just pushes back the understanding of how it could have formed by purely naturalistic processes. All right, so this is a statement from Dr. John Sutherland. Over 100 years. Okay, wait, maybe I should read the quote first, but I will just go ahead and read it right now. So, over 100 years after abiogenesis was proposed, scientists have yet to create a functional protein from purely inorganic molecules under prebiotic conditions. Scientists have yet to create a fully functioning RNA molecule from purely inorganic molecules under prebiotic conditions. So just understand, like, they've been working very, very hard on this, and they have made some progress in some cases, but in these experiments, they're making them very finely tuned, which actually is evidence for some sort of intelligent design. Because if you constantly need intervention in these experiments, and then they're not just happening on their own, then you're showing the need for some sort of some sort of intelligent design to step in and coordinate things. So anyway, and then fun fact, no living cell has ever been created in the lab from only non-living components. And you'll see on headlines like they created a cell in the lab. No, what they'll do is they'll use an already living cell, right? And then they'll change the DNA out or whatever it may be. Cloning kind of happens like that. Basically, you're switching out that DNA of an already living cell. So they're not just creating a cell from scratch. That has not happened. And it would be extremely difficult to make it happen. And I'm sure you would need a lot of intelligent design to make it happen from those scientists.
Yeah, definitely.
So do you want to read this quote real quick?
Yeah, I was trying to read it and find a way to summarize it. But, understanding how life on Earth might have originated is a major goal of Origins of Life chemistry. To proceed from simple livestock molecules and energy sources to a living system requires extensive synthesis and coordinated assembly to occur over numerous steps, which are governed only by environmental factors and inherent chemical reactivity. Demonstrating such a process in the laboratory would show how life can start from inanimate. If the starting materials were irrefutably primordial, and the end result happened to bear an uncanny resemblance to existent biology, for what turned out to be purely chemical reasons, albeit elegantly subtle ones, then it would be a complete recapitulation of the way that natural life originated. We are not yet close to achieving this end, but recent results suggest that we may have nearly finished the first phase. Well,
first phase. But yeah, he is an abiogenesis researcher and he believes in it. But at least he's conceding that they haven't even finished the first phase of it. So, yeah. And it says the prebiotic synthesis of building blocks to which we have devoted so much of our time only corresponds to a small increase in the complexity of the system and to no increase in its aliveness. This is a humbling thought, he said.
Yeah, you only become more and more aware of how complex it is and how more impossible it is rather than getting to some solution. That's it.
Exactly.
So the next segment, we'll talk about a positive case for intelligent design.
Positive case. So now we're going to be giving some positive evidence. So a lot of people try to say, "Oh, well, you just believe in intelligent design because that's just the God of the gaps, right? We don't understand it, so you're just putting God in that gap." No, what I'm doing and what we're doing is taking what we know about things that are intelligently designed and looking at the cell. And we could say, yeah, we see a lot of things in the cell that look like other things that we've designed or that have indicators of an intelligent designer. So it's not a God of the gaps. We're making a claim based on what we're actually observing. All right, so here's some indicators of intelligent design. Complex and specified information. Organized, meaningful complexity. We got the irreducible complexity as well, where all parts are essential for function. Purposeful arrangement, features, designs for specific goals. Fine-tuning, precise calibration of parameters, information-rich features, detailed intelligence, associated information. We got the specified complexity. We got the adaptation of means to ends, where all parts work together for a clear purpose. And we have the creation of new or novel information. Novel functional information suggesting intelligence. So yeah, here's some good indicators of something that is intelligently designed. All right, so we're going to move into a couple cases of irreducible complexity. Do you know if this was the start of your segment?
Yeah, it is.
Okay.
I'll go ahead and take it from here. So irreducible complexity is basically just stating that like one thing can't be formed without the other, that the whole system doesn't work without an essential component. So the system of like DNA, RNA, and tRNA, making ribosomes, like it all is like super integrated network. So each component's dependent on the other for its function, or else there'll be death or disease. And now I'm going to show you how the ribosome is a complex molecular machine that requires 50 translated, preformed proteins to facilitate the protein synthesis and support the argument for intelligent design. So one thing that the ribosome does is that it translates. And why is it called translation? It's translating between two completely different languages, right? With different spellings and different systems of units. And these have to be like matched up like perfectly for them to work. So there's 3 nucleotides in the DNA or RNA, and that corresponds to 1 amino acid. And there's 64 possible codons from the mRNA, and it must be matched up to 20 different, specific amino acids.
Right.
And you can use this little like wheel here whenever when you have like a sequence of mRNA and you're trying to find out which amino acids they're going to make.
So you basically have, I just want to make a comment. Like, so you basically have these codons of 3 nucleotides from the RNA that then gets translated to 1 amino acid. So you have to have that translating system already there.
And here's like a basic picture of that little translating sequence. This is the ribosome right here, and this is a strand of mRNA. And the picture up here is showing something similar, just like that there could be multiple ribosomes on a single strand of mRNA. And we're going to talk a lot in detail about the tRNA right here, the transfer RNA, which is the one that's actually translating between the codons of the mRNA and then picking out which amino acid we need to use. Okay, so this highly specific translator, it's translating between 2 languages, like I said. And then there's like the amino acid attachment. We'll talk about that more. Okay, so the tRNA, first we should take a look at its structure. It has, so what does it do, right? We already kind of went through this, that it reads the mRNA. It attaches amino acids to one another, and they all carry their own specific amino acid. And
so you're saying there's like 20 different tRNAs, one
yeah,
for each amino acid?
Yes, this acceptor stem here.
So not only do we have to have one of the translators, we got to have 20 of them because there's 20 amino acids.
Yeah, like we're going to get to it next, the aminoacyl transfer charging right here. Perfect. Okay, so like you're saying there's different ones, and that's because of their attachment site. Because of this necessary enzyme right here called aminoacyl tRNA synthetase. It's going to recognize this attachment site, and it's going to combine, the one specific attachment site to it. The amino acid it needs. So this enzyme, like I said, it combines each amino acid to the necessary tRNA, and if it doesn't work properly, it can recognize that and cut it off and try to replace it with the right one.
Correct.
And I was going to say, so this is just another example of a necessary preformed protein. And
20 of them.
Yeah, 20 different ones, right?
Yeah.
And then
crazy.
This is a good time to talk about the specific structure of the tRNA itself. There's like a T arm and a D arm, and they have like essential components of each that are necessary for folding and necessary for it to, function right. And also you can see here, energy is needed, right? So even if you're imagining some, self-replicating tRNA situation, it still has to be in a perfect environment like Taylor's been describing. So the whole ribosome together, right? That's that ball we were talking about, how this whole process is going through. Protein synthesis happens in the ribosome, and ribosomes are molecular machines made of 2 very large RNAs and over 50 preformed proteins. There's 3 different sites in the ribosome. There's an A site, a P site, and an E site. The A site is where the next charged tRNA enters, and then there's the P site where the amino acid from the next amino acids being added onto the chain, basically. The elongation site, it's called. And then the E site is like an exit site where it's kind of resting for to leave and restart the whole process. And I guess I was going to describe here, like this picture up here describes that elongation situation going on. There's a whole like little process going on here, and this is the protein being produced. And as you can see, this molecule itself is extremely complex, requiring over 50 proteins to function. If any one of those proteins are not there, then disease can occur. The most common one,
And it
or
won't function.
yeah, it won't function right.
Yeah, but what's the disease? Sorry.
But there's a disease, Marie Charcot tooth. It's like a really well known because of its a failure at this step.
Mm.
And then I just threw this in because I was like, in case you thought, oh, well, that's it, So this is
Yeah.
just pretty much what I've said before.
How many proteins does it have in the RNA? Is it like 30 or something?
The RNA?
The
Like
ribosome.
the ribosome.
Yeah, the ribosome.
So like over 50. And
Over
that's in
50?
that's in a bacterial ribosome.
So a bacterial ribosome has over 50 proteins. So this is
yeah,
needed.
yeah.
This is needed to create a protein and
Yeah.
translate from the RNA to the proteins.
Exactly.
Yeah.
And I mentioned that because they're a little bit different. You can see there's like the small and large segment of the ribosomes are a little bit different from one another.
Hmm.
You need these elongation factors for the I think the tRNA to bind to the ribosome.
Yeah, it's a lot.
Yeah. Let's see. Is there anything else? Release factors. I mean, there's just a lot of stuff going on. Okay, so even the minimally functional translation system is inherently complex, requiring ribosomes, tRNAs, AARNAs, that's the charging enzyme, and translation factors. Aminoacyl tRNA synthetases, which attach amino acids to their corresponding tRNAs, are essential for translation. Yet these enzymes themselves require a functional translation system to be synthesized.
Yeah, synthesized. Yeah, so we have they're trying to propose like a solution to this. Like, how could this mediator have came about to mediate between RNA and amino acids and proteins? And basically they were saying, oh, like there could be stereochemistry where like the amino acids like the RNAs and they're on them, and then they start forming this like relationship with one another, and then they could transcribe them. But
Are they not?
first of all, when are they still gonna have like the translation information between them? Like, that's the first thing. And then the second thing is, remember earlier how I was talking about that Maillard reaction where the synthesis of nucleotides and amino acids, their conditions have to be separate. So the amino acids will react with the sugars like a ribose, which is a component of RNA, which would deplete the available ribose for RNA synthesis.
This is used commonly in medicine to test for fructose in the urine, which is like a biochemistry issue in children.
Wow, that's cool. So yeah, that couldn't have happened.
Exactly.
And now we're looking at
the irreducibility, right? So we're looking at molecular machines.
That's exactly. Like, we have these amazing machines in the cell that are required for essential purposes. We got the flagellum up here. We have DNA replication down here. Look how amazing that process is. It's amazing.
And it happens so fast. This
Yeah,
is like slowed down times 100.
This is extremely slowed down. But yeah, they're absolutely amazing. And then we have the mitochondrial ATP synthase. All right, so we have specific and irreducible complexity. Remember earlier I was saying that was one of the indicators of an intelligent designer. All right, so Charles Darwin stated in The Origin of Species, if it could be demonstrated that any complex organ existed which could not have possibly been formed by numerous successive slight modifications, my theory would absolutely break down. So that's amazing. And I argue that we found many systems like this.
Yeah, it's broken down. Trash. They're in the trash.
Yeah, so a system with several interacting parts, if just one part were to be removed, the system would not function. So that's basically irreducible complexity. And there's many cases of it. The flagellum being one of them. And one of the common rebuttals is, oh, well, there's this other flagellum that we have that required less stuff, but actually, even in that flagellum, it has irreducible complexity as well.
Yeah, I can actually explain. I think what happens is that there's this is like a rotary motor, right? This thing, I think it can go up to like 100,000 RPM. It's reversible and it functions at nearly 100% energy efficiency. Okay, but what they were talking about was that there's these like kind of like an attack flagellum. I don't know the exact name for it, but it's how like one bacteria or something will go like attack another one. It's just totally different. It's more of like a syringe that extends outwardly, and it has no rotation purpose.
But I'm sure even that thing has irreducible complexity.
I'm sure it's wild.
Yeah.
And I just want to mention these things up here. Like this is kind of this is in the core of the flagellum, and I think it's super cool. It's something that is kind of related to Taylor's research here. There's all these different little parts, like these little arms. These little connector arms are called stators, and there's dozens of proteins that are in this thing, and if any one of them are removed, it doesn't function.
Yeah, it doesn't function. So this is kind of like what the flagellum tail up here at the top would look like if you just cut it in half. So yeah, these are just showing like the microtubules that will radiate through it, and yeah, it's an amazing feat. All right, so here's ATP synthase. This was also very similar to man-made motors. So ATP synthase and man-made motors share key similarities. Both utilize rotational mechanisms with a central shaft rotating within a stationary housing, resembling the rotor and stator designed in electric motors. They generate torque to perform work. They are modular in structure and adaptable, such as that ATP synthase can operate in reverse. Additionally, they convert energy with remarkable efficiency, except ATP synthase actually achieves a near 100% efficiency as compared to man-made motors. So these are truly amazing. And here's a little schematic of it up here, just kind of showing how it works. So this is how we generate energy in our cells required. All right, so intelligent design and DNA, one of the things is complex specific information. So DNA's organized code acts like digital instructions. And over here is actually a diagram of from inside of the cell, from the mesoderm and ectoderm, basically giving feedback. So they have these like feedback loops already incorporated, and basically feedback on which genes to form more of, which genes to form less of, and the cell, how it resembles like
Yeah.
computer chips, for example, many parts of the cell.
This is basically telling the cell where it's at in development and helping it decide what cell it's going to become. I
Yeah,
see.
there's a lot of feedback and regulatory
I
mechanisms.
think
in
this
the
is
cell.
like my section.
Yeah,
to
yeah,
kinda-
you can go ahead.
And so in a similar manner, like we're looking at how there's like informational hierarchy and stuff. So we've talked about how like DNA, makes genes, ribosomes, and the genome. And then there's like molecules that then can form organelles, mitochondria, and cells. And cells make tissue, organs, organ systems, organs. Same way your neurons make neural circuits, brain regions, and it makes up your whole central nervous system. But I put these pictures here because I think it's just so cool that, we look at these computer chips and we're like, man, this is super complex. And there could be, a lot of integrations and like switches and a lot of decision making happen. Well, look at this, circuit that you have going on in every single one of your cells. All these things are necessary for you to break down what's like glucose over here, go through glycolysis, the citric acid cycle, and make all these different biomolecules that you run on. And even some of these, well, I just want to say that like these things don't just happen for no reason. Like, and it's really hard to see from your screen, but there's all these little blue words written out on here, and they're all necessary enzymes.
Yeah, so everything is integrated, and we can see that in things that we've created, like the circuit board and computers. So then we look in the cell and we start seeing all this integrated and all these integrated systems, and we're like, wow, that resembles something like what we've made. So, and all of that is necessary. Like they all have to be working in conjunction with one another and giving feedback to one another, et cetera, et cetera. So it's just really impressive.
Yes, indeed.
All right, so here's a paper.
Yeah, this
go
is
ahead.
cool. You talked about like the computational architecture of the cell. The complexity of the cell and its corresponding analogies to contemporary computing systems runs deeper than simply DNA. The cell as a whole has also been viewed as a computational machine similar to multiprocessing clusters with a combination of a centralized instruction set and distributed computing elements. The channels of information transfer and transformation in the cell have direct analogies in communication theory and computational algorithms. And
That's
so,
amazing.
like the processing system would be like a gate and circuits, or like the amino acids as basic elements.
Yeah, you got the information code. We have the binary codes in the computers. We got the quaternary code with the 4 base systems in the DNA. You got the primary memory. That would be like the gate in a computer, and the ribonucleotide as a basic element engineered in circuits. So there's so many different examples of just similarities between computer systems and our cell and DNA. Let's see if there's any more cool ones I can find. Is there any more you
Yeah,
want to
no,
share?
I
This is just awesome.
I want to spend more time reading this paper. It's
Yeah.
so cool.
That's awesome, man.
Yeah.
And you got like the data, the basic computing structure. You got the data path in a computer, and then you got the proteins, which carry out the functions. And yeah, it's amazing. All right. So is abiogenesis a viable theory? All right. So theories must have a coherent explanation to whatever phenomena it is trying to explain. And abiogenesis completely lacks this. Okay. There is no coherent explanation to the start of life. And many times I see them appeal to the future. right? Like, oh, well, maybe sometime in the future we'll figure it out, right? But that's actually, fallacious. And ironically, the more we learn about the cell, the more unreasonable abiogenesis becomes. We're actually learning about all this circuitry in cells and informational hierarchy that is needed. So I feel like we're going in reverse. And then personal incredulity, just because you can't understand it, just because you don't want to believe intelligent design or whatever, doesn't make it false, right? Just because you can't understand it, that doesn't make it, something that's not viable. So I find a lot of scientists have this where they don't want to say it's intelligently designed, so they'll just go with the contrary. But you should definitely look at all the evidence first. And it is viable intelligent design, or is intelligent design viable? All right, so Occam's razor prefers explanations with fewer assumptions. So intelligent design has one. Okay, and think about all the assumptions they had to make for the first cell that they're still trying to figure out in abiogenesis. Whereas intelligent design postulates one abstract concept, right? So they have one postulate in intelligent design versus hundreds, dozens in abiogenesis. All right, so these prebiotic experiments that they're actually doing are proof of intelligent design because they're having to intervene in these experiments. And so they model the need for information and interaction needed in the origin of life. So whenever they're constantly stopping the reaction, adding new reagents, and controlling the pH levels, they are showing the need for an intelligent designer and something that's intervening in this process. And so, as I mentioned earlier, the intelligent design is not a God of the gaps. It is based on what we do know about where information comes from. And we can observe the attributes of something that is intelligently designed and then see those same attributes in the cell. So we're going off of what we do know about things that are intelligently designed rather than, oh, well, we don't know what happened, therefore it must be God. No, we're not doing that at all. All right, and I'm going to end it with this quote. This is Dr. George Wald, and he actually won a Nobel Prize in 1967 in medicine. And he said, wait, yeah, that's the wrong quote. That's the wrong quote. But basically, he was saying that he doesn't want to attribute God to the origin of life research. And so he's willing to believe something that's scientifically impossible over the idea of God. So yeah, that's about it. And intelligent design isn't rejected on scientific standards, but by philosophical ones. And we should definitely not do that. We should definitely hold it to scientific standards. If science is showing us that this looks like it was intelligently designed, that's what we should go with. All right, and thank you guys for listening. I wanted to give acknowledgments. Thank you to Daniel for helping me with the tRNA slides, and to Sam, Andrew, and Peter for helping me with organization. Thank you to Alan and Galatrax for moderating. Thank you to Ether Cause and Earth Awakenings for giving me a platform, and I'm open for any questions, corrections, or rebuttals.
Thank you.
If anybody's still there.
Oh, I think people are putting hands up.
Okay.
Okay.
Should I end the stream?
Well, you can just like call them up because we'll keep the slides up or whatever.
Can you join as a listener so I can see who's asking questions?
Oh, sure.
Yeah. Okay, I'll keep the screen. Yeah, I'll just keep it on in case anybody has a question about a slide.
Yeah, excellent presentation, guys. Appreciate the shout out, by the way.
Yeah, no
I'm not
problem.
sure if Omnivore wants to come up and ask his question about the flagellum. He said he has a question about. Oh, I'll let him ask it. There he is. Go ahead and bring him up.
So yeah, so I have heard about the flagellum being irreducibly complex, and I've heard counters to it. You said there's something similar to a flagellum that needs less pieces, but it's also irreducibly complex.
Yeah, so it's actually not a flagellum, but it kind of looks like it in shape, but it doesn't function as a flagellum. So what it is, and I wish I knew the name of it. It basically, it's like an attack syringe essentially for one microorganism, I believe a bacteria, to then consume another bacterium. And so it's like more like a straw that extends out, and it actually like fires out really quickly, leading me to believe that it's probably, like Taylor said, there's more evidence of irreducibly complex structures in that thing. But
Yeah.
it is
It was just-
fundamentally.
What are the
Flagelum-
walls? Okay, well,
What's that?
what
Yeah,
are
I
the?
didn't decide on that. I just kind of threw that in as a side point where
Mhm.
some people say, "Oh, well, they have this other thing like a flagellum that has less parts to it, therefore irreducible complexity is invalid." But like,
So
necessarily.
okay, well, I
I
mean,
don't have a slide on that one, but
so
I just
so Beastocles was kind of a jerk to me when I pointed this out, and I want to make it real clear to him that if you're gonna say that I'm just being incredulous about something and then go off on me, Beastocles, you might want to hold your tongue next time. So about about that particular issue, right? That's a particular piece of evidence that we could look at. Well, I mean, you were kind of spicy there, Beastie. So I think it's all fine.
Right.
But you can. So when you're talking about the flagellum itself, I'm making it about you. You're the one who jumped in on that. If you don't want me to make it about you, just stop typing. And I've said all I need to. So the are you talking about the macro structures that are created by it, or are you talking about the base
just the whole
proteins?
just the whole rotary system as a whole.
Yeah,
Like in
that's
a motor. Like think about
If I remove, okay, you can go to the next. You can go to the next one, Geo. I didn't start the drama. If you don't want it, then
I can't hear anybody else except for Omni.
Yeah,
they're typing in the chat. They're typing in the chat to me as we do this.
No, it's okay.
So if you remove proteins from the DNA that creates the flagellum, can you still get a flagellum out of it?
Yeah,
My
I think
understanding is you can.
Yeah, I think they've actually tried this before and they found it to not be functional. But that was something I meant to add more about in my slides. I just haven't had time. Like it took me so long to put together this presentation. I couldn't add absolutely everything, but I can hit home on that if I present again on exactly about the flagellum in particular.
Yeah, because there's some I'll put a paper in the chat for all the people who are incredulous.
Yeah, and it's not really just the flagellum. It's many of the molecular machines in the cell. This one's just interesting because it's actually like a rotary motor. And so if you think about a motor and how it functions, if you remove parts from that motor, it's not going to function. So then I guess the question is but
I don't know that. So there's two things, right? There's the DNA code that creates the flagellum.
Mm-hmm.
And then there's the flagellum itself, right? So those are two different things. So are you saying that one of them's irreducibly complex and the other one isn't?
No.
Oh, they're both are most likely.
The flagellum gets the information from the DNA, so they're not like different. Basically, the proteins that make up the flagellum are encoded by the DNA. So you're saying basically if you knock out a part of that DNA and knock out one of those proteins, will it still work? I mean, yeah, I don't know. And I think that they've done a paper on that and tried to alter the flagellum and it did not function from my memory, but I don't have a paper on hand to give you.
So I posted one. I just posted a link to one that I think they were able to do that. So I have to check and make sure that's the right paper. But yeah, I would question that example.
What gene did they knock out in that paper exactly? Like which protein did they knock out? Shall I ask? Which part of this flagellum did they knock out? I'm just curious.
So they knocked out COG 1463.
I think that's a species.
Is C
I don't even see that
uppercase
on
or
here.
lowercase?
Uppercase.
COG 1463. And so what did that encode for? Was that the protein?
It's one of them.
What does it
Interacting proteins is MOTB. I'm not an expert on this. The analysis, if the answer is I have to have a degree in bacterial biology in order to understand whether or not this is a case of irreducible complexity, you might be right. But if that's the case, that's kind of the problem.
Okay, well, if you could just let me know what part it knocked out or whatnot, that's cool. But
And what was the COG part again? COG 1?
COG what?
I put the link in the chat at 30 minutes past the hour.
Okay.
So they have a whole. It's more generally. It's not about knocking out proteins and having the flagellum work or not. That's not what they were
I
doing on
think
there.
they're
This
looking
is
at a different species, right? A different species flagellum. Isn't that
they're
correct?
looking at flagellas. I don't know. I mean, is there a
I
mobility is
think they're
achieved by
addressing-
most bacterial species by the flagellar apparatus. It consists of dozens of different proteins with thousands of individual subunits. So it looks like they're just talking about flagellums in general. Is there one particular flagellum you're thinking of?
Like,
No.
are you asking us like which flagellum are we referring to?
If you're referring, well, she said they're talking about a different species of flagellum. I don't.
I think
You
that
know.
you're addressing the same thing that I addressed, which is that they said there's like a different type of flagellum altogether, but that type would still be, have irreducible complexity. And the purpose of irreducible complexity is like, okay, so if we don't have like how would these start forming this rotary motor if it didn't have an end in mind? Like there's no goal in mind. So why would it continue to move towards that goal if it had no function? So if you look at this system right here, the flagellum.
I'll tell you right here, what they modified was this thing over here on the side, the stator. I knew it. I knew that if you knock something out, I looked it up. What the gene you were talking about is the stator. Okay.
Well, that's just one. That's one of several.
Yeah.
I mean, they
It's
did a
not
whole
e-
bunch.
it's like saying like what there's probably
We took the door off a car and it still
exactly
worked.
right. Like how
Okay, so it's not the motor; it's the whole car.
yeah, like even if you take the transmission off the motor, it's still going to run. You know what I'm saying?
Right.
And
But,
so
but wait a minute. Originally, we were on the motor, right? Or we were just saying
Yeah.
it's pieces of a motor, not it's pieces of the whole car. So, what piece can you take off of a
Well, we're talking about the essential rotor in the center, like the rotary motor, and the whole thing as a whole. Because how would any of this start to form if it has no end in mind? That's the real point here. Like, how would it even start to build if it couldn't function?
So, I've heard that argument too. I don't know. I don't.
That's what it is.
I don't
That's
know
the
that.
argument.
Yeah, but I don't.
Point is, let me give you the point of this, right? The point is that there's no purpose for it to continue to evolve something that has no function. And without all these pieces, even if you want to knock out, the door of a car, without all the pieces of the motor of the car and essential components, it's still not going to go. You know what I mean?
I don't think you can go.
There's no reason for it to continue to evolve something that has no function without all the pieces there.
So is it
You know what I mean?
is it possible that a partial flagella confers some advantage?
Well, think about it. Like, is there a partial motor, right, that's missing crucial elements? Can it still function? That's the question.
Yeah, but we went from a motor to a car with missing a door. So I don't know.
The thing you're talking about is this purple bar over here. It still would be able to push like energy through it and stuff and still form. You're talking about something that is associated with it that might help it be more stable in the membrane.
Yeah, but they
go ahead and knock out one of these components of it and make it and watch it work. You understand what I'm saying?
Okay. All right. Yeah, but they did a bunch. That was just the first of several.
Then you should have then tell me which ones they knocked out.
The MOB, the thing on the very most outside.
Yeah. But it's not even really about that. The whole
FLHA, FLGE2, FLIE. I mean, all of these show this paper is not about knocking out proteins. That's not what it's about. It's about whether or not the flagellum can function with modifications to those or removal of those proteins. If a protein doesn't function, what happens to the flagella? So they didn't. It's not attempting to disprove your.
Did they find it was functional? What was the results of the study?
Yeah,
the result was that it was still functional. Yeah, from what I understand.
Okay. Well,
from the MO.
Hey, can you give me the name of that paper really quick? I'll look up.
I'll protein network of bacterial motility.
I sent you both the paper in Discord DMs. You both have it in your DMs on Discord.
Oh, thank you so much. Yeah, I'll check it out, and hopefully I can give you a response before the live stream ends.
Okay.
Okay.
It was
Galatrax.
Thanks, Beast. That was good.
I'm glad you got your question out.
Oh, I did.
We got Robert V Utah up here. Did you want to ask a question as well?
Yeah.
Yes, I want to ask a question. The name of the space is called scientifically dismantling the myth of what? Making vehicles? What myth are we?
Abiogenesis.
Biogenesis.
Abiogenesis. Yes.
Abiogenesis. I see.
Were you here for the presentation?
Yeah, presentation and contribution. Yes, sir.
Yes, indeed. So we're just talking about how life is super complex and it's awesome, and it was designed by an incredibly intelligent God because he made all these super cool components that we get to appreciate. You want to
I don't think intelligence really sums up what God really is capable of. It's
Yeah.
beyond that, you know.
Yes,
So,
indeed.
right, you know, because God is the Alpha and the Omega. So He, all
Amen.
things He has created. Um, you know, and what I take from that as a Christian is that we can't limit God. You know, many Christians try to limit God. For example, this big discussion about the shape of the earth. Many Christians are limiting God's power, saying that He created a little flat plane for us to just live on with a dome over it.
Robert, we're trying to keep the presentation to stuff regarding the subject of abiogenesis and
Yeah.
their arguments surrounding it. So if you have any questions about that, go for it, brother.
Yeah, no. I was just responding to the gentleman's question there.
Yeah.
So how do you guys see, understand abiogenesis? Like,
So what we're doing, so
how do you guys
abiogenesis,
see that?
like I think the word abiogenesis means life from non-life, and so we've been arguing, really just absolutely demolishing abiogenesis by showing that the positive case for intelligent design. Like, why should we be very reassured that life shows evidence that it was intelligently designed? And so we sort of systematically went through like a bunch of different things. But if you have a question about any of those things, we'd love to talk about them.
The thing is that remember, God created us from the dust, man from the dust, right? Women were created from a little bit more than that, right? So what's dust? What's the dirt? Dust is just non or living matter. So so the abiogenesis does not eliminate intelligent design. I think that's the confusion, right? So we have to be careful as Christians what we're communicating to people and so on.
Okay.
So God knows He's the creator. You
So,
understand me?
I kind of like am picking up what you're putting down, right? And it's sort of the idea that maybe you're saying that, God grabs the dust and that breathing life into it makes life. But like there's many people who don't want to talk about God and are terrified of him, frankly. So they employ all their possibilities to come up with some scientific reason as to try a scientific reason to ignore God. But what we're more saying that we know for a fact that it couldn't have happened without God. Does that make sense? Like,
Okay, knowing. Okay, so Christians are, scientists are going to always push back on that. But the deal, what we have to understand as Christians is that, um, you know, God's timetable and God's time frame, because the scripture even talks about it. We're on His timetable. So, the 7 day creation is not 7 of our days. It's a process that God is going through. And when they talk about evolution again does not eliminate God. It doesn't, right? So we have to understand that the creator process and timetable is different from ours. I know it's a controversial thing that I'm saying, but we have to not limit God. We
Well,
have to not do that.
then don't say he can't do it in a day or 7 days or 6 days. He did it in 6 days. So we're not going to limit him because he did incredible things in each day and in 6 days in total, right? And so we're definitely not trying to limit him. And another thing is that you're right. I believe that if someone was to somehow come up with some evidence that evolution is true, that also does not eliminate God. Because I believe that God intervenes and gives us every breath and is always doing things. In fact, I'll tell you something. I'm so glad we're on this slide. I read a whole paper about how this ATP synthase right here, it doesn't make any sense. Like the energetics of putting like an ADP with one phosphate group, combining 2 other phosphate groups to it to make an ATP, an adenine triphosphate, doesn't energetically make any sense. Okay, so this process is going on. We know that ATP has to be made. However, it's as if it wants to function in reverse. But what it's doing is it actually is creating energy. And so this obviously challenges like a big portion of the biochemistry textbook. But I'm arguing that like God is driving our ATP synthesis like every second. He's your breath of life.
No, absolutely. Come on, he wouldn't do that. You really thinking that God is in all of us turning our wheels? No, come on, no.
Oh yes, 100%.
No, absolutely
100%.
not.
Yeah.
Absolutely not. But if
So
he
what
was doing that, then we do not have free will. He
Oh no, no, no. I'm
specifically,
just saying
he's
giving you the ability to
specifically
have free will.
independent us.
Listen, I'm not telling you he's controlling everything you do and you have no free will. What I'm saying, he's giving you the opportunity to have free will. He gave you life. He
Right,
gave you a
right,
mind. He gave you the
right.
ability to have free will. I'm not,
Yeah, but he's not. There's no way he's in our mechanics, biology doing it. No, he
Look,
already did that.
I'll use your words. You said not to limit God. Thank you,
Right.
sir.
Yeah. You. But that's a limitation. Our
Don't limit
Okay.
God.
Sounds good.
Okay, so I was just reading this paper that Galatrack sent me from Omni, and basically they were able to find 164 genes that they knocked out that completely inhibited the function of the flagellum. So, if anything, this gives more credence to intelligent design because they showed multiple examples of how if you knock out one protein, it does not function.
So they gave an example of knocking out 160 different ones,
And it stopped working.
and it stops working. So
That's exactly my point.
The level of irreducible complexity really just got larger. You see
Yeah.
what I'm saying? You see how you dig and you dig and you dig. Oh, we're looking for answers. The only answer you're going to find is that irreducible complexity is seen in all sorts of things, and it becomes even harder and harder to fight for abiogenesis.
I got a question real quick. Could you go back to the pages with the paradoxes because there are so many of them, and I just think that's like those are so amazing to look
I
at.
know, I
Okay,
love this. I'll just start from
asking for a friend.
I guess. Like, so we have the time, natural selection, the tRNA, circular synthesis, ribozyme, membrane,
Oh,
Elgin.
yo. Sorry, I cut that dude off in the middle of his thing. I thought I was up here to unmute to announce what was happening or whatever, but I was saying if you guys don't want to continue like a meta conversation about, the limitations of God, but like this is the actual physical mechanisms that are put forward as the explanation, and you guys are dismantling them to a degree to where it's not even a tenable position to say like, oh, but if we like invoke unlimited time, maybe God, like, no, God didn't make it retarded, right? Like this is a very clear set of instructions that have to, like, it would have to be spontaneously done or not. It's not an overtime issue. We're not limiting God. But anyway, like we're looking for specific, refutations to the mechanisms that are put forward rather than like appeal to infinite possibility through God. So anyway, you guys are welcome to, anyone's welcome to come up and ask questions and stuff, but
Any
please don't
comments,
be
questions?
endlessly circular.
Yep.
Yeah.
Good point, Alan. Honest. Yeah. Just what's your guys' most like in all the paradoxes? Which one do you guys think is like the strongest one against abiogenesis? out of all of those you presented?
It would be easier to say if they actually had a coherent theory.
Yeah,
like,
there's no.
like there's no it's just the incoherence of their different theories and the situations that would have to happen for each.
The most common one is the time. I hope I
The
didn't cut
time
you off, but
paradox, yeah.
you'll
Time
hear so many
paradox,
people,
right?
even our grandparents, are like, "Oh, well, you never know what happens over billions of years." I'm like,
Yeah.
"Yeah, a bunch of things degrade. What you ought to know that."
Yeah.
You know, um, I don't know. She asked, "What's our favorite one?" I mean, I don't go. There's so many. I guess I'll take back to like
I
the
like the information, like the tRNAs, like how even if we hypothetically had all these amino acids there and we hypothetically had all these nucleotides that were in the right chirality, suitable for life, and even if we had everything, like what's going to translate between them? And like they're working so hard just for these organic molecules. And like even like if you did get one, you have to have all these systems in place to do something with it. That's the point. Like everything has to be working in coherence and already there for it to be functional. And they're like really trying extremely hard just to make like a functional RNA just from inorganic compounds. And like assuming that they even did, like then what? You have to have all these other things to make it work and do something, you know? So that's one of the biggest points that I have.
Gotcha. We do have RG the artist here. He did want to ask a question. RG, you got the floor.
Hey, thank you very much for that wonderful speech. That was nice. I learned a lot tonight. Um, so Taylor, I was wondering, with how our environments are always feeding us light information, and I'm wondering through your studies about how maybe the continuing um feed from our outside environment, how does that affect and influence the cells in DNA?
Wait, I'm sorry. Like, can you repeat your question? Like, so how does the outside environment affect our cells?
Yeah, like the light, like through the skin, and then through the rest of the processes, I guess, like from the moonlight or like the magnetism or different spectrums from the sun, I guess, UV and stuff like that.
How does it influence life?
Yeah, like the continuing information from your natural environment. Yeah. I guess is that
Well, the sun and the light is actually very important. Push full screen. Sorry, I'm trying to get this right quote up that I was trying to end with.
Was it the George Wald one? Yeah, do that one.
Yeah. It'd be nice if we can make it a little bigger. But just to answer your question, I was just gonna say sunlight is very important and key for like things like what's
And
it
making
called?
vitamin D.
D. And
Yeah.
then remember that researcher that you were telling me about that actually says like the light that you eat in is more important than what
Oh
you
yeah,
eat.
Jack Cruz.
Jack Cruz, he's
Yeah.
awesome. Yeah, Shane sent me a video from him recently.
Awesome.
And yeah, it's amazing. Like, and also light, like biophotons. That's like a kind of a new area of study for biology. Like we're seeing that we have these biophotons that could be sending information within the neurons because the information, how quick it gets sent within our neurons is like incredibly quick. And some people say that the chemical theories, right, even though they are playing a role, is not fast enough to translate and transmit the information that we need, you know, and whenever we're functioning.
Mhm.
So basically, there's theories about biophotons that are being conducted through like the microtubules, which is what I study, to basically transmit information in the brain.
Yeah, another thing about that, and this is quite a while since I read this paper, but I'll just try to keep it short. They basically took slices of different species brains from mice to like maybe dogs to like monkeys, and were shining light through the same sized thickness of the slices of brains. And as you were basically like moving up in species, like these more intelligent species were absorbing more of the light, so the light was more redshifted. And what they're saying is they simultaneously were looking at the glutamate receptor. So glutamate receptor is stimulated on a neuron and makes it depolarize and will then continue an action potential to another neuron. Well, the point is that light will activate the glutamate receptors. Okay, so one neuron can carry light down it like a fiber optic cable and then shine on the next neuron in the same way that it action potentials.
And it's like
and
transmitting information.
Yeah.
I've heard about that. That's really interesting. So, things like that just show
That's
how much
awesome.
more amazing the cell is than we know.
Yeah, yeah,
And here
and
we
I
go.
would encourage. I was going to say I would encourage you guys to look into also bioplasma. There's a lot of good studies going on with that as well. Yeah.
That's awesome.
What is that? Bioplasma?
I'm not sure.
Well, like
Could you tell?
your auric field extends out from your internal bioplasma, which basically is the building blocks for your neurons and neural pathways.
Wow.
Bioplasma. Check it out.
Let me That's interesting. Thank you for sharing. I just wanted to read this quote from earlier. So this is George Wald, and he actually won a Nobel Prize, and he studied at Harvard. And he said there are only two possible explanations as to how life arose: spontaneous generation arising from evolution, or a supernatural creative act of God. There is no other possibility. Spontaneous generation was scientifically disproven 120 years ago by Louis Pasteur and others, but that just leaves us with only one other possibility: that life came as a supernatural act of creation by God. But I cannot accept that philosophy because I do not want to believe in God. Therefore, I choose to believe that which is I know is scientifically impossible: spontaneous generation leading to evolution. George
At
Wald.
least he's honest enough to admit that there's
Yeah,
a little bit
exactly.
of a philosophical bias there.
We have to give him credit for that. But this is what we're dealing with here. I feel like a lot of people just have cognitive dissonance when it comes to intelligent design. Even if everything is pointing to intelligent design, it's just glaring in your face. They're like, no, it couldn't have been. Has to be another way. Can't believe in that. You know?
Well, yeah, what are the implications if there is a God?
Exactly.
Jeez.
Exactly.
And if anyone else has any questions, just raise your hand and we can bring you up and you can ask any questions. How long do you guys think you're going for? How long would you guys like to go for?
We can go however long you guys want.
Yeah, I mean, as long as y'all have questions and want to talk about it, or just discussions, comments, like
Any feedback,
anything we could have done better or whatever.
And Dan here is in medical school, so he could probably answer a lot of health questions if you have any.
Have some random questions we can talk about it together.
Right now, you are currently streaming Discord. I have a question.
Yeah,
When did you guys
Yeah.
first start questioning abiogenesis? What was the moment for each of you?
We stopped streaming our computer.
Like,
so
when did you first start questioning abiogenesis?
So I first hold on. Can we stop the stream?
Yeah.
I don't even know how to do that. Oh, you just pushed that one. Okay. Cool. So I first started considering abiogenesis kind of like it's always kind of been in the back of my mind, like how life got created. And then as I mentioned, I asked my 5th grade teacher how life arose, and then she said, oh, well, chemicals must create more chemicals. So I was like, that was ridiculous. I mean, the way she was answering, I knew that she didn't know what she was talking about, and she was kind of thinking about it herself, like, that is an interesting question. But yeah, I've always thought about it, and it's just been kind of common sense to me that something like that could not just happen by accident. What about you?
I'm really trying to think about it, and it's been an evolution, like my understanding of abiogenesis and my personal feelings about it probably really started like in biology 2 class, but man, it really like was solidified in biochemistry, because like I showed that biochemistry map, and I remember like learning about well, like in biology 2, learn about a bunch of different body parts and stuff, and like how the body functions. And I was like totally amazed by it, thinking like, how is this all like how could all this be an accident or anything? But then in like biochemistry, you learn that these chemical reactions, they have like delta G constants or whatever, where the reaction doesn't occur on its own unless you have this enzyme, which is a preformed protein. So then you learn that the enzyme is this protein that is only made in the cell through this complicated process that we talked about.
Yeah.
And so I said, well, shoot, if the thing needs itself to function, then it can't come about on its own.
Yeah, like you need an energy input for a chemical reaction to happen, but these chemical reactions do not naturally happen. So that's why we have enzymes that come and lower that activation energy for the reaction to happen. But without the enzymes, nothing's going to happen. That's why they're having to invoke these crazy circumstances and prebiotic earth and conditions in order to make these reactions happen. They don't happen naturally. And if they do, they usually just organic molecules just degrade over time and
Mm-hmm.
don't form anything. Like in nature, if we're just looking out into nature, like aside from any life, like considering prebiotic Earth, everything moves towards non-life. Everything will degrade and have entropy naturally. You're having to jump over so many hurdles in order to make this theory work, as you can see. It's not something that happens naturally.
And I'm guessing the answers you guys got during your education was not satisfactory, so you decided to look a little deeper, and then
Mhm.
you came up with this awesome presentation. To
Thank
which,
you. Yeah, right.
did you guys have any more comments on the paper that Omnivore suggested?
Well, Omni suggested that paper. Yeah, I went and looked at it, and basically, they found 164 genes that whenever they knocked out, the flagellum did not work.
Yeah, like I commented on that
Oh,
slide and I said there was like a dozen or a couple dozen proteins that if you knock out, it doesn't function. Well, it turns out it's 160
Yeah,
genes instead.
even more.
So if you
that
knock
is
out
even
any
worse.
one of those 164 genes, it ceases to function.
Yeah.
Ah, gotcha.
Okay, so a little bit of self-debunking then. Did you guys know that abiogenesis comes from the prefix a, not bios, life,
Yeah.
and
Yeah.
genesis, birth, origin, or creation? So not life origin, not life birth, not life creation.
Yeah, and that actually contradicts like with the laws of biology. Like, there was a funny scenario that happened when I was in school. Basically, we had like a biology quiz, and one of the questions had to do with basically the laws of biology, how life only comes from living cells, right? Life only comes from life. And then I put, no, that's wrong because abiogenesis. And then I was incorrect. And then I was like, but wait, didn't y'all say life came from non-life on early Earth? And then she was like, yeah, I guess, like, and gave me the point back. But I think maybe she wasn't a fan of it either.
Oh, you think she didn't entertain abiogenesis, but she may have
Yeah.
had to for the curriculum?
I know from my experience, a lot of my biology professors, like kind of under the table, didn't support abiogenesis, and some of them openly saying that God created all this. No way.
Yeah.
You know.
So it's like it's not the best idea, but it's what we've got for now. It's a work
Exactly.
in progress idea, and then
It's
But
I
they're
had
moving
some comments
backwards.
on your presentation too. I
Yeah.
think first I want to say thank you guys for presenting. You two formed a watertight premise. The conclusion follows. It tracks. It's sound. You guys have proven that abiogenesis as an explanation for the origin is untenable, and it's not a sound argument. It doesn't track. It's not sound. And then, as far as the positive claims section goes, which is the second section, usually the question is, what would it take to falsify your premises? But your entire presentation is covering that, so it seems like there'd be too much. There'd have to be way too many things to falsify it. The odds are stacked against the abiogenesis. So the
Exactly.
so-called scientific theory of abiogenesis sounds more like special pleading, appeal to possibility, a la philosophical bias.
Exactly. Yeah. They're
Takes a lot
just
of
moving
faith.
they're moving in reverse. And it would one of the things that would make me question if abiogenesis was tenable is if they were able to actually put organic compounds together and get life. But
Mhm.
or even get like some moving towards life. But what we're seeing, like I said, is they're like they are a lot of times they're incompatible, and you have to have special, enzymes moving these reactions and compartments in the cell to separate these environments they need to be in. But without that, you just have them all mixed together, and that is detrimental. But that's what would have been needed. So that's why they have to make all these special cases of this had to form here and this had to form here, and then how do you get that all to come together is the question.
I really like the graphic that you guys showed, which had that mall arrow that points to it. You know, at the mall it says "You are here," and it points to that that slice at the bottom.
Yeah.
We did have a hand, but it went away.
I see.
I do have
Yeah.
one more question, if that's okay.
Oh yeah, for
Um,
sure.
so it's kind of a broad question, and it's for both of you that did the speech here. So as creationists, what are y'all's future plans in this field that you're, you know, talking to us about, and what what would be the area of most impact that y'all would like to work on next together? to make an impact and to make a statement, maybe, as
Well,
creationists.
hmm I was actually talking to Dr. Brian Miller the other day because he was looking through my PowerPoint, and they do have creationist researchers, and I would love to participate in that, but it doesn't really affect researchers like currently, right? Because like most of the time you're just in the lab working on your own research, following the scientific method, and anything that's like following the scientific method and like showing that something's real is like tenable, right? And I don't have a problem with that. But it's the crazy claims that are like have these just such impossibilities and that are so far-fetched that they're like positing as fact to people that I have the problem with, because we should be utilizing the scientific method to, find out what's true and what's not.
Yeah, that's true.
Yeah, absolutely.
And I guess
Ben,
on my
did you?
Yeah,
Yeah,
like
go ahead.
so I'm currently training to become a physician, and hopefully will one day become a general surgeon. And I think that understanding that we're all intelligently designed and we're all, brothers and sisters, helps you to show compassion and love for each other in a way that you wouldn't do otherwise. And so just I hope that it helps me like become more loving and impactful to others in a positive way.
And a lot of physicians are actually like that.
Yeah.
They believe in intelligent design. There's a lot of scientists who also do. They just get kind of pushed to the side because like there's a overarching, materialism that's hovering over science, and we should definitely try to remove that and just look at everything empirically and say, what is the evidence actually pointing to, even if it's not what we want to conclude. So we should be more open-minded in that fashion.
Awesome. So stay optimistic, and thank y'all for doing this again. One more question, last question, and I'm gonna fall back and land the plane here. So I don't know if y'all have done much research in like the forces of like the weak forces or the strong force, like and magnetism. I'm sure that plays a role, like as far as holding these structures together, which are super tiny, right? But super intricate as well. So, do y'all think about that much, or have any opinions on that? Like the
Well,
role it plays?
I'm not sure if I can really comment on the role that it plays, but I know one thing as I was thinking about the like different like reactions that happen between like functional groups and stuff, and I mean I guess I just don't know how it works in this conversation, but there's like certainly so many different conditions have to be present for like all the different necessities of life to come about,
Yeah.
and different, I don't know, that's it.
Yeah, so I was just gonna say like the magnetic field, like birds and stuff like that can pick up on it, and like they do like migration and stuff, and they hypothesize that there's these like magnetic proteins in their brain that allows them to, based on the magnetic field, be able to know
Or in
which
their
direction to go.
eyes.
In their
It's
eyes?
in their eyes, yeah.
That's
So maybe they
interesting.
can like, see a
That's
compass.
weird. That's weird. Or see like some type of magnetic field lines, or
Yeah.
not lines, but just like something. I don't know how it functions. I don't think scientists necessarily know completely either, but yeah, magnetism, very interesting. So I know in terms of birds, they can utilize magnetism, but I don't know about humans. I'm sure there's some magnetic field that permeates around us in some way. And also like the quantum properties of microtubules, they have like frequencies. And they found that if you alter these frequencies or expose them to different frequencies, they can actually either stabilize or degrade. So like you could be prone to that in your environment.
All right, we have Fundy up. Floor is yours if you have a question, comment, etc.
Hey, thanks, man. Yeah, great stuff, guys. I don't know. I got here a little late, I think, but did anyone ask you the age-old question, what came first, the chicken or the egg yet?
Yeah, we covered that one.
Yeah.
Okay, look, I got a,
Maybe the RNA, what's between a chicken and an egg?
I always
The
answer.
chiclet.
I've always answered this question with the chicken because nothing. Any animal's always going to lay the egg of itself, right? So if you're talking about a chicken egg, the chicken has to lay a chicken egg, and so like
What did God create first?
Exactly.
Probably the chicken.
you know,
Yeah.
I hope that answers the
Ab-
question. I mean, you need to have the
Absolutely.
C4, you know.
Right. Just poof, fully formed chicken. Of course, every chicken came from an egg, and every chicken or every egg came from a chicken. So it's yeah, quite the conundrum. I got a more serious question though. Um, do you guys see anything in your research that points towards life actually having some beginning, some amount of years ago? Or is it on the table that life, like living beings, is something that could have always has been and always will be, and didn't necessarily have a first created thing, but basically just always living things have always been around?
Where on Earth or in the universe?
Where wherever. Just I guess the
Well,
universe.
there's been some whatever whatever your definition of life is, then yeah, there's something conscious that's always been here. I believe in an immovable mover, right? There's something that had to move, that had to create, and that was never created. Because if you continue to ask, oh, well, who created the creator? That's like the infinite regression paradox, and you never have a cause to the effect. And so I think that there's always been some sort of life per se, and some sort of intelligence, and something that designed this place. But in terms of biological life, I'm not sure how long that's been around. I don't know if we were the first humans ever created or not. Biblically, yes, we would be, but just depends.
Cool, thank you. Last question. I don't know much about it. Just wondering if you've ever heard of I forget the guy's name, but he's famous for this concept of the morphogenetic field. If that's of any interest to you guys.
Hmm.
I'm gonna have to look into that.
Do you know anything about it? Like
morphogenetic.
Just the basics. You guys would probably recognize the name of the guy who like coined the term, but it's kind of the idea that either in the DNA or something that there's basically like an electromagnetic field that is then filled in with the flesh, that like there's sort of like a predestined shape that guides like the cells to sort of fill out like coloring within the lines of a children's book or something, and that the lines are sort of yeah, I'm not sure I can put it much better than that. I'm not too much of an expert myself on it. Just wondering if you guys had heard about it, but yeah, check it out maybe.
It seems like it might be kind of related to like energetic healing or something. Like you have like energy fields like in your body, and it drives development and stuff. Like in the development of the embryo. Well, we didn't even really talk about that, but man, that's amazing. You really want to get, shaken up. Go like look at how we develop in the womb. It's incredible.
Someone posted in chat another paper called "Magnetic Bones in Human Sinuses." So I sent that to both you two. You can check it out.
That's dope. Thank you for sharing. Cool. All right. Well, I've really enjoyed being here. Does anybody else have a question before we head out?
A quick one, yeah. How do you guys know you wouldn't be happier believing in random cosmic coincidence?
Well, I mean, would you be happier thinking that you came from a monkey? Because I wouldn't. I can't stand the whole monkey man theory. It's ridiculous.
Okay.
I'd be lost. You see, people having a purpose is essential for is very important for being fulfilled. And it's we should all be glad to know that there is strong evidence that you were designed and built for a purpose, and you were designed by an incredibly intelligent God who loves you. I think that should only benefit everyone, right? I don't understand why you would be opposed to that.
Because people just want to act however they want.
Yeah, probably.
Yeah,
Thank you
well,
guys. Presentation was awesome. I
yeah, thank you,
love
Scott.
watching you guys. You have great synergy. I like how you guys go back and forth. It's awesome. This is all really great. Thank you guys.
Yeah, thanks for
Thank
having
you.
me. And if anybody has any questions, you could just DM them to me, and we'll get back with you. Thanks for being here.
Thank you.
All right, so I think we can wrap it up there. Gio, if you want, we could move everyone down to the live stream channel now.
husband for Valentine's Day.
Yeah. It's okay.
Okay.
Oh, did either of you guys get each other flowers for Valentine's Day?
I just said Yep.
Wait, is Galatrax in here?
Yeah,
Are those
I'm in here.
orchids?
Yeah, they're orchids. The whole orchid plant.
That's
Yo,
amazing.
orchids are like one of the most unique flowers out there. I love them so much. Good
Yeah.
choice.
I told both of you guys to get the other